Welcome to the video lecture for Unit 4, Module 9.3. In this one, we're going to discuss acid-base balance. As always, here are your learning objectives. Please be sure to reference these when studying for both your lecture quiz and the unit exam. Let's go ahead and get started. So we introduced the topic of pH and the chemical nature of acids, bases, and buffers in 168. Table 23-7 reviews key terms important to the discussion that follows. The pH of the body fluids can be altered by adding either acids or bases. In general, we can categorize acids and bases either as strong or weak. Strong acids and strong bases dissociate completely in solution. For example, hydrochloric acid, which is HCl, a strong acid, dissociates in solution by the reaction HCl dissociates to hydrogen plus chloride. When weak acids or weak bases enter solution, a significant number of molecules remain intact, so dissociation is not complete. In other words, if you place molecules of weak acid in one solution and the same number of molecules of a strong acid in another solution, the weak acid will release fewer hydrogen ions and will have less effect on the pH of the solution than will the strong acid. Carbonic acid is a weak acid. At the normal pH of the ECF, an equilibrium exists and the reaction can be diagrammed as follows. Carbonic acid dissociates into hydrogen ions and bicarbonate ions. The pH of body fluids reflects interactions among all the acids, bases, salts in solution in the body. The pH of the ECF normally remains within narrow limits, usually 7.35 to 7.45. Any deviation from the normal range is extremely dangerous. Changes in hydrogen concentrations disrupt the stability of the plasma membranes, alter the structure of proteins, and change the activities of important enzymes. You could not survive long with an ECF pH below 6.8 or above 7.7. When the pH of blood falls below 7.35, acidemia exists. The physiological state that results is called acidosis. When the pH of blood rises above 7.45, alkalemia exists. The physiological state that results is called alkalosis. Acidosis and alkalosis affect virtually all body systems, but the nervous system and the cardiovascular system are particularly sensitive to pH fluctuations. For example, severe acidosis, or pH below 7, can be deadly because 1. Central nervous system function deteriorates and the person may become comatose. 2. Cardiac contractions grow weak and irregular, and signs and symptoms of heart failure may develop. And 3. Peripheral vasodilation produces a dramatic decrease in blood pressure, potentially producing circulatory collapse. The control of pH is a homeostatic process of great physiological and clinical significance. Both acidosis and alkalosis are dangerous, but in practice, problems with acidosis are much more common. This is so because normal cellular activities generate several acids, including carbonic acid. There are 3 general categories of acids in the body. They are 1. Fixed acids, which do not leave solution. 2. Organic acids, which take part in or are produced by cellular metabolism. And 3. Volatile acids, which can leave the body and enter the atmosphere through the lungs. Carbonic acid is an important volatile acid in body fluids. It forms spontaneously from carbon dioxide and water in body fluids, but this reaction proceeds much more rapidly in the presence of the enzyme carbonic anhydrase. This enzyme is found in the cytoplasm of red blood cells, liver, and kidney cells, parietal cells of the stomach, and many other types of cells. The partial pressure of carbon dioxide, or the PCO2, is the most important factor affecting the pH in body tissues. Most of the carbon dioxide in solution is converted to carbonic acid, and most of the carbonic acid dissociates into hydrogen ions and bicarbonate ions. For this reason, the partial pressure of carbon dioxide and the pH are inversely related. In other words, when carbon dioxide levels increase, additional hydrogen ions and bicarbonate ions are released so that the pH decreases. Recall that pH is a negative exponent, so when the concentration of hydrogen ions go up, the pH values goes down. In contrast, as carbon dioxide leaves the bloodstream and diffuses across the alveoli, the number of hydrogen ions and bicarbonate ions in the alveolar capillaries decreases and the blood pH increases. We'll consider this process, which effectively removes hydrogen ions from solution in more detail later in the module. To maintain acid-base balance over long periods of time, your body must balance gains and losses of hydrogen ions. Hydrogen ions are gained in the digestive tract and through metabolic activities within the cells. Your kidneys eliminate these ions by secreting hydrogen into urine. The lungs eliminate them by forming water and carbon dioxide from hydrogen and bicarbonate. These sites of elimination are far removed from the sites of acid production. As the hydrogen ions travel through the body, they must be neutralized to avoid tissue damage. Buffers and buffer systems in body fluid temporarily neutralize the acids produced by normal metabolic operations. Buffers are dissolved compounds that stabilize the pH of a solution by adding or removing hydrogen. Buffers include weak acids that can donate hydrogen and weak bases that can absorb hydrogen. A buffer system in body fluids generally consists of a combination of a weak acid and the anion released by its dissociation. The anion functions as a weak base. In solution, molecules of the weak acid exist in equilibrium with its dissociation products. In chemical notation, this relationship is represented by HY dissociates to H plus Y. Adding hydrogen to the solution upsets the equilibrium. As a result, additional molecules of the weak acid form, removing some of the hydrogen from the solution. The body has three major buffer systems: phosphate buffer system, protein buffer system, and carbonic acid bicarbonate buffer system. Each has slightly different characteristics and distributions. As we look more closely at these buffer systems, remember that they provide only a temporary solution to an acid-base imbalance. To preserve homeostasis, the hydrogen that these systems capture must ultimately be either permanently tied up in water molecules through the removal of carbon dioxide by the lungs, or removed from the body fluids through secretion by the kidneys. For that, respiratory and renal mechanisms are needed, as we'll later see in the module. The combination of buffer systems and these respiratory and renal mechanisms maintains body pH within narrowed limits. The phosphate buffer system consists of the anion H2PO4, which is a weak acid. The operation of the phosphate buffer system resembles that of the carbonic acid bicarbonate buffer system, which is discussed below. The reversible reaction involved is dihydrogen phosphate dissociates into hydrogen ions plus monohydrogen phosphate. The weak acid is dihydrogen phosphate, and the anion released is monohydrogen phosphate. In the ECF, the phosphate buffer system plays only a supporting role in the regulation of pH, primarily because the concentration of HCO3 far exceeds that of HPO4. However, the phosphate buffer system is quite important in buffering the pH of the ICF. In addition, cells contain a phosphate reserve in the form of the weak base sodium monohydrogen phosphate. The phosphate buffer system is also important in stabilizing the pH of urine. The dissociation of sodium monohydrogen phosphate provides additional hydrogen phosphate for use by this buffer system. Protein buffer systems depend on the ability of amino acids to respond to pH changes by accepting or releasing hydrogen, that is, by acting as a base or an acid. Such molecules are said to be amphoteric. If pH increases, the carboxyl group of the amino acid can dissociate, acting as a weak acid and releasing a hydrogen ion. The carboxyl group then becomes a carboxylate ion. At normal pH of body fluids, the carboxyl group of most amino acids have already given up their hydrogen ions. Proteins carry negative charges mainly for that reason. However, some amino acids, notably histidine and cysteine, have R groups, or side chains, that will denote hydrogen ions if the pH increases outside of the normal range. Their buffering effects are very important in both the ECF and the ICF. In a neutral solution, an amino acid carries both positive and negative charges. This state is called a Zwitter ion. The term is derived from the German meaning the hybrid ion. If pH decreases, the carboxylate ion in the amino group can act as weak bases and accept additional hydrogen ions, forming a carboxyl group and an amino ion, respectively. In free amino acids, both the main structural chain and the side chain can act as buffers. In a protein, most of the carboxyl and amino groups in the main chain are tied up in peptide bonds. Only the exposed amino group and carboxyl group at either end of the protein are available as buffers. So most of the buffering capacity of proteins is provided by the R groups of the amino acids. Plasma proteins contribute to the buffering capabilities of blood. Interstitial fluid contains extracellular protein fibers and dissolved amino acids that also assist in regulating pH. In the ICF of active cells, structural and other proteins provide an extensive buffering capability. That buffering prevents destructive changes in pH when cellular metabolism produces organic acids, such as lactic acid or pyruvic acid. The protein buffer system can also help stabilize the pH of the ECF because exchange occurs between the ECF and the ICF. For example, when the pH of the ECF decreases, cells pump out hydrogen out of the ECF and into the ICF, where intracellular proteins can buffer them. When the pH of the ECF increases, pumps and plasma membranes move hydrogen out of the ICF and into the ECF. These mechanisms can assist in buffering the pH of the ECF, but the process is slow. Hydrogen ions must be individually transported across the plasma membrane. For this reason, the protein buffer system in most cells cannot make rapid, large-scale adjustments in the pH of the ECF. The situation is somewhat different for red blood cells. These cells, which contain approximately 5.5% of the ICF, are normally suspended in the plasma. They are densely packed with hemoglobin, and their cytoplasm contains large amounts of carbonic anhydrase. Red blood cells have a significant effect on the pH of the ECF because they can absorb carbon dioxide from the plasma and convert it to carbonic acid. Carbon dioxide can diffuse across the RBC membrane very quickly, so no transport mechanism is needed. As the carbonic acid dissociates within an RBC, The bicarbonate ions diffuse into the plasma in exchange for chloride ions, a swap known as the chloride shift. Hemoglobin molecules buffer the hydrogen ions. In the lungs, the entire reaction sequence proceeds in reverse. This mechanism is known as the hemoglobin buffer system. The hemoglobin buffer system is the only intracellular buffer system that can have an immediate effect on the pH of the ECF. The hemoglobin buffer system helps prevent drastic changes in pH when the plasma PCO2 is increasing or decreasing. Body cells generate carbon dioxide virtually 24 hours a day. Exceptions are red blood cells, some cancer cells, and tissues temporarily deprived of oxygen. As we've seen, most of the carbon dioxide is converted to carbonic acid, which then dissociates into a hydrogen ion and a bicarbonate ion. The carbonic acid and its dissociation products form the carbonic acid bicarbonate buffer system. The primary role of this buffer system is to prevent changes in pH caused by organic acids and fixed acids in the ECF. The carbonic acid bicarbonate buffer system consists of the reaction introduced in our discussion of volatile acids. So carbon dioxide and water dissociates into carbonic acid, which dissociates into hydrogen ions and bicarbonate ions. Because the reaction is freely reversible, a change in the concentration of any one participant affects the concentrations of all the others. For example, if hydrogen ions are added, most of them will be removed by interactions with HCO3, forming H2CO3 or carbonic acid. In the process, the bicarbonate acts as a weak base that buffers the excess hydrogen. The H2CO3 formed in this way in turn dissociates into carbon dioxide and H2O. The lungs can then excrete the extra CO2. In effect, this reaction takes the hydrogen released by a strong organic or fixed acid and generates a volatile acid that can be easily eliminated. The carbonic acid bicarbonate buffer system can also protect against increases in pH, although such changes are rare. If hydrogen ions are removed from the plasma, the reaction is driven to the right. The PCO2 decreases, and the H2CO3 dissociates to replace the missing hydrogen. The carbonic acid bicarbonate buffer system has three important limitations. 1. It cannot protect the ECF from changes in pH that result from increased or decreased levels of carbon dioxide. A buffer system cannot protect against changes in the concentration of its own weak acid. An equilibrium exists among the components of this buffer system. Thus, in this system, the addition of excess hydrogen from an outside source would drive the reaction to the left, but the addition of excess carbon dioxide would form H2CO3 and drive the reaction to the right. The dissociation of H2CO3 would release hydrogen and HCO3, decreasing the plasma pH. 2. It can function only when the respiratory system and the respiratory control centers are working normally. Normally, the increase in PCO2 that occurs when fixed acids or organic acids are buffered stimulates an increase in the respiratory rate. This increase accelerates the removal of carbon dioxide by the lungs. The efficiency of the buffer system will be reduced if the respiratory passageways are blocked, or blood flow to the lungs is impaired, or the respiratory centers do not respond normally. This buffer system cannot eliminate hydrogen and remove the threat to homeostasis unless the respiratory system is functioning normally. 3. The ability to buffer acids is limited by the availability of bicarbonate ions. Every time a hydrogen ion is removed from the plasma, a bicarbonate ion goes with it. When all the bicarbonate ions have been tied up, buffering capabilities are lost. Problems due to lack of bicarbonate ions are rare for several reasons. First, body fluids contain a large reserve of HCO3, mostly in the form of dissolved molecules of the weak base sodium bicarbonate. This readily available supply of bicarbonate is known as the bicarbonate reserve. When hydrogen ions enter the ECF, the bicarbonate ions tied up in H2CO3 molecules are replaced by HCO3 from the bicarbonate reserve. Second, additional HCO3 can be generated by the kidneys. In the distal convoluted tubule and collecting system, carbonic anhydrase converts carbon dioxide within tubular cells into H2CO3, which then dissociates. The hydrogen ion is pumped into tubular fluid in exchange for a sodium ion, and then the bicarbonate ion is transported into the peritubular fluid in exchange for a chloride ion. In effect, tubular cells remove HCl from peritubular fluid in exchange for sodium bicarbonate. Buffer systems can tie up excess hydrogen, but they provide only a temporary solution to an acid-base imbalance. The hydrogen ions are merely rendered harmless. They are not eliminated. To preserve homeostasis, the captured hydrogen must ultimately be either permanently tied up in water molecules through the removal of carbon dioxide by the lungs, or removed from the body fluids through secretion by the kidneys. The underlying problem is that the body's supply of buffer molecules is limited. Suppose that a buffer molecule prevents a change in pH by binding a hydrogen ion that enters the ECF. That buffer molecule is then tied up, reducing the capability of the ECF to cope with any additional hydrogen. Eventually, all the buffer molecules are bound to hydrogen, and further pH control becomes impossible. The situation can be resolved only by removing hydrogen from the ECF, thereby freeing up the buffer molecules, or by replacing the buffer molecules. Similarly, if a buffer provides a hydrogen ion to maintain normal pH, homeostatic conditions will return only when either another hydrogen ion has been obtained or the buffer system has been replaced. The maintenance of acid-base balance thus includes balancing hydrogen gains and losses. This balancing act involves coordinating the actions of buffer systems with the respiratory mechanisms and renal mechanisms. These mechanisms support the buffer system by 1. Secreting or absorbing hydrogen, 2. Controlling the excretion of acids and bases, and when necessary, 3. Generating additional buffers. As we've noted, it is the combination of buffer systems and these respiratory and renal mechanisms that maintains body pH within narrow limits. Respiratory compensation is a change in the respiratory rate that helps stabilize the pH of the ECF. Respiratory compensation takes place whenever body pH strays outside of normal limits. Such compensation works because respiratory activity has a direct effect on the carbonic acid bicarbonate buffer system. Increasing or decreasing the rate of respiration alters pH by decreasing or increasing the PCO2. When the PCO2 increases, the pH increases because the addition of carbon dioxide drives the carbonic acid bicarbonate buffer system to the right. When the PCO2 falls, the pH increases because the removal of carbon dioxide drives that buffer system to the left. Chemoreceptors of the carotid and the aortic bodies are sensitive to the PCO2 of circulating blood. Other receptors located on the ventrolateral surfaces of the medulla oblongata monitor the PCO2 of the CSF. A rise in PCO2 stimulates the chemoreceptors, leading to an increase in the respiratory rate. As the rate of respiration increases, more carbon dioxide is lost at the lungs, so the PCO2 returns to normal levels. Conversely, when the PCO2 of the blood or the CSF decreases, the chemoreceptors are inhibited. Respiratory activity becomes depressed and the breathing rate decreases, causing an elevation in the PCO2 in the ECF. Renal compensation is a change in the rates of hydrogen and bicarbonate secretion or reabsorption by the kidneys in response to changes in plasma pH. Under normal conditions, the body generates enough organic and fixed acids to add about 100 milliequivalents of hydrogen to the ECF each day. An equivalent number of hydrogen ions must be excreted in urine to maintain acid-base balance. In addition, the kidneys assist the lungs by eliminating any carbon dioxide that enters the renal tubules during filtration or diffuses into the tubular fluid as it travels towards the renal pelvis. Hydrogen ions are secreted into the tubular fluid along the proximal convoluted tubule and the distal convoluted tubule and the collecting system. The ability to eliminate a large number of hydrogen ions in a normal volume of urine depends on the presence of buffers in the urine. The secretion of hydrogen can continue only until the pH of the tubular fluid reaches about 4.0 to 4.5. At that point, the hydrogen concentration gradient is so great that hydrogen ions leak out of the tubule as fast as they're pumped in. If the tubular fluid lacked buffers to absorb the hydrogen ions, the kidney could then secrete less than 1% of the acid produced each day before the pH reached its limit. To maintain acid-base balance under these conditions, the kidneys would have to produce about 1,000 liters of urine each day just to be able to keep pace with the generation of hydrogen in the body. Buffers in tubular fluid are extremely important because they keep the pH high enough for hydrogen secretion to continue. Metabolic acids are being generated continuously. With these buffering mechanisms, the kidneys cannot maintain homeostasis. The three major buffers involved are the carbonic acid bicarbonate buffer system, the phosphate buffer system, and the ammonia buffer system. Glomerular filtration puts components of the carbonic acid bicarbonate buffer system and the phosphate buffer system into the filtrate. Tubule cells, mainly those of the PCT, generate ammonia. The hydrogen ions generated may be pumped into the lumen in exchange for sodium ions, individually or together with chloride ions. The net result is a secretion of hydrogen, accompanied by the removal of carbon dioxide from the tubular fluid of the tubule cells in the ECF, and the release of sodium bicarbonate into the ECF. As tubule cells use the enzyme glutaminase to break down the amino acid glutamine, amino groups are released as either ammonium ions or ammonia. The ammonium ions are transported into the lumen in exchange for sodium in the tubular fluid. The ammonia, which is highly volatile and also toxic to cells, diffuses rapidly into the tubular fluid. There it reacts with a hydrogen ion, forming ammonium. 1. This reaction buffers the tubular fluid and removes a potentially dangerous compound from body fluids. The carbon change of the glutamine molecules are ultimately converted to bicarbonate, which is co-transported with sodium into the ECF. The generation of ammonia by tubule cells ties up hydrogen in the tubular fluid and releases sodium bicarbonate into the ECF, where it contributes to the bicarbonate reserve. These mechanisms of hydrogen secretion and buffering are always functioning, but their levels of activity vary widely with the pH of the ECF. Acidosis, or low blood pH, develops when the normal plasma buffer mechanisms are stressed by excessive hydrogen ions. The kidney tubules do not distinguish among the various acids that may cause acidosis. Whether the decrease in pH results from the production of volatile, fixed, or organic acids, the renal contribution remains limited to 1. The secretion of hydrogen, 2. The activities of buffers in tubular fluid, 3. The removal of carbon dioxide, and 4. The reabsorption of sodium bicarbonate. Tubule cells thus bolster these capabilities of the carbonic acid bicarbonate buffer system. They do so by increasing the concentration of bicarbonate ions in the ECF, replacing those already used to remove hydrogen ions from solution. In a starving person, tubule cells break down amino acids, yielding ammonium ions that are pumped into the tubular fluid. Bicarbonates help to keep buffer ketone bodies in the blood and carbon chains for catabolism. When alkalosis, or high blood pH, develops, 1. The rate of hydrogen secretion by the kidneys declines. 2. Tubule cells do not reclaim the bicarbonates in tubular fluid. And 3. The collecting system transports bicarbonate into tubular fluid while releasing a strong acid, or HCl, into peritubular fluid. The concentration of bicarbonate in blood decreases, promoting the dissociation of bicarbonate and releasing of hydrogen ions. The additional hydrogen generated by the kidneys helps return the pH to normal levels.